Our next speaker is Mehmet. He's from the Medipol University in Istanbul. So we switch pathogen again. We come to Leishmania, and we will hear about new emulsions for delivery of water-insoluble drug candidates against Leishmaniasis.
Thank you for the nice introduction. First of all, I would like to thank to the organizers. I'm very pleased to be here each time, each CLINAM. My name is Mehmet Hikmet Üçışık. I'm from Istanbul Medipol University, and the title of my talk is Solid Lipid Emulsions for Delivery of Water-Insoluble Drug Candidates Against Leishmaniasis. Leishmaniasis is considered as a neglected parasitic disease, and it is widely seen in more than 98 countries worldwide. These countries also include Southern and Eastern European region, as well as Mediterranean intercontinental region. And annually, more than 1.5 million people are affected worldwide, where the death rate reaches up to 100,000. There are different forms, including cutaneous, mucocutaneous, and visceral forms present, and out of which the visceral leishmaniasis is the most severe one and may become fatal if untreated. And in its progression, macrophages and other cells of reticuloendothelial system serve as host as well as replication site for the parasite. The parasite of visceral leishmaniasis is resistant to lysis by lysosomes and specifically reside inside the macrophages of spleen and liver. Visceral leishmaniasis is caused by intracellular protozoan parasites, in particular Leishmania donovani and Leishmania infantum. And Leishmania have two main life cycle stages. The motile-flagellated promastigote, which is present in the sandfly vector, and intracellular non-flagellated amastigotes, which are present in the mammalian host cells. The life cycle begins with an infection of the female fly after a blood meal on Leishmania species infected vertebrate hosts. And amastigotes proliferate in the midgut of sandfly vector and differentiate in the foregut into metacyclic promastigotes. Once delivered to a vertebrate host by the bite, promastigotes attach to phagocytic cells and taken up by the macrophages. And inside the macrophages, the parasite containing parasitophorus vocals fuse with the lysosomes and form phagolysosomes, wherein promastigotes differentiate their form into intracellular afflagellated amastigote form. And then the proliferation of the parasites continue and the disease become more and more serious. Oops. And the treatment of leishmaniasis, in particular visceral leishmaniasis, requires chemotherapy. And World Health Organization's recommendations for treatment of VL caused by Leishmania infantum in the Mediterranean basin, Middle East, and Central Asia is Ambisome, a liposomal formulation of amphotericin B as the first-line drug, and pentavalent antimonials, including meglumine antimoniate and sodium stibogluconate as the second-line drug, and amphotericin B as the third-line drug. Paramomycin and miltefosine are the other two options, but they are not highly recommended due to their less efficacy and higher toxicities. And whereas we do have some chemotherapeutic agents, these therapies are usually unsatisfactory due to the emergence of drug resistance, dose-limiting toxicity issues, and limited efficacy. And even more, the therapies may not always be available for undeveloped countries due to the route of administration and high cost. As we have also heard from in the previous talks, the Leishmaniasis is a larger problem, in particular in undeveloped countries, and we have to provide cheaper solutions and cheaper therapies for them to overcome this disease. And these limitations address the need for novel anti-leishmanial chemotherapeutic agents and novel nanoformulations. Addressing these requirements, in the recent years, there are several new drug candidates which are found to be potentially effective against Leishmaniasis. Among them, I would like to underline bis-naphthalimidopropyl BNIP derivatives. And the aim of our study is to develop a novel anti-Leishmania drug nanoformulation with higher efficacies and minimize adverse effects using emulsions as the nanocarrier system and bis-naphthalimidopropyl derivatives as the drug agent. To start with, emulsions describe lipid-based nanocarrier systems composed of an internal solid fat core surrounded by phospholipid multilayers. The lipophilic load, as you can see in the sketch, can be localized both inside the inner fat core as well as within the phospholipid layers. The key feature of this nanoformulation is that the fat core is in a solid or liquid crystalline phase rather than oil in a fluid phase. This feature in particular distinguishes emulsion from emulsions as well as allows the encapsulation of higher amounts of lipophilic compounds within the solid matrix. They differ from solid lipid nanoparticles by the lack of any presence of surfactants. Nonetheless, when formulated with phospholipid layers and triglycerides, SLNs have exactly the same composition. So, why do we prefer emulsions? First, the emulsions offer high load capacity for hydrophobic substances such as BNIP. And stabilized by phospholipids, emulsions do not require any presence of surfactants as stabilizing agent, which makes them highly compatible. Controlled drug and solid lipid matrix enables the release of the drug gradually upon uptake into the cell. And least but not the least, accumulation in the organs of the reticuloendothelial system, RES, instead of the kidney. Emulsion largely reduces the toxicity of the drug, and they improve the anti-Leishmaniasis efficacy as parasites are also located in these organs and tissues. Several studies have been already reported in the literature on different types of diseases, including viral infections as well as autoimmunity. Among them, there are also amphotericin B formulated emulsions present for macrophage-targeted delivery. And our active pharmaceutic ingredient is, as I have just mentioned, BNIP derivatives. BNIPs are formulated by two naphthalimido groups linked with a natural, linked with a natural polyamine chain, which determines actually the point of diversity. And BNIPs possess particular sirtuin inhibition, up to 17-fold selectivity for parasitic sirtuin. Their mechanism of action occurs over inhibition of NAD+ dependent deacetylase activity. Besides its antiparasitic activity, it has been also shown that BNIPs are also effective against cancer. And they strongly interact with DNA, and naphthalimide planar aromatic rings of BNIP intercalate between DNA-base pairs. They distort the DNA backbone conformation by interfering with DNA-protein interaction, and they induce cell cycle arrest and apoptosis. The drawback of this drug candidate is its low water solubility and shown toxicity on macrophages. Therefore, to address these two drawbacks, our study outline is first of all the synthesis of novel BNIP derivatives originating from the most active found BNIP derivative, which is BNIP DAOCT, with a linker chain composed of 8 carbon length, and formulation of BNIP DAOCT loaded emulsions. And at the end, screening of this formulation for its biological activity on both Leishmania infantum parasites as well as infected macrophages. To start with the characterization, BNIP DAOCT formulation has a mean size of around 541 nanometer. It has a negatively neutral average theta potential, and it enables the loading of BNIP up to 0.4 milligram per milliliter. And due to the autofluorescence property of BNIP duct, encapsulation could be also seen by confocal microscopy. And as we have in our cell culture studies on promastigotes, we have seen that the IC50 value of BNIP duct is similar to the values in the literature. The IC50 value of BNIP duct was found as 0.84 micromolars, whereas the encapsulation of BNIP duct into the emulsions have improved the effectivity of the drug with a decrease in IC50 value of 0.59 micromolars. And the free emulsion formulation does not show any significant toxicity against promastigotes at concentrations as high as 30 micromolars. Those two bars are for emulsion formulations without the drug inside. And then we have also done the toxicity studies with the healthy macrophage cell line, RAV 264.7, and we have seen the toxicity on the studies with BNIP duct free form molecules, whereas the encapsulation within the emulsions have decreased this toxicity, and the EC50 values was obtained for BNIP duct was 5.3 micromolars, whereas it became 17.1 micromolars. So by only looking to these EC50 values and the effectivity against promastigotes, we can see that the selectivity of BNIP duct, which was calculated as 6.3, has been increased to nearly 30. And free emulsion formulation does not show any significant toxicity on macrophages at all, even at high concentrations as 30 micromolars. And as the last cell culture study, we have infected our macrophages with Leishmania infantum parasites. On the left-hand side, you see the phase contrast image. And in the right-hand side, you see the multi-channel image where Leishmania is colored by green with PKH67 dye, and infected macrophages is labeled with red with PKH26 dye. And the treatment of Leishmania infantum infected macrophages, the studies on treatment of Leishmania infantum infected macrophages has revealed out that both BNIP duct and BNIP duct emulsion decrease the infection rate up to 20%. So 80% of the infected macrophages have been killed or have been cured actually. And the infection ratio of Leishmania infantum infected macrophages were determined using flow cytometry, where we have also seen the percentage of infected macrophages at negative control, nearly 69%. When treated with free emulsions, it remained the same. And after treatment with 10 micromolars BNIP duct free molecules, it dropped to 27.26%. And after treatment with 30 micromolar BNIP duct emulsion, we could achieve 22%, an increase to 22%. So overall emulsion formulation improved BNIP duct efficacy on infected macrophages. And in conclusion, BNIP duct has a potential drug candidate for Leishmaniasis with an IC50 value of 0.84. And the incorporation of BNIP duct into emulsions, the biological activity of BNIP duct has been improved with a decrease in IC50 value, nearly 0.6 micromolars. Indicating the safety of emulsion formulation, BNIP duct emulsion showed a 29-fold selectivity for promastigotes And lipid-based emulsion formulation give promising data to address solubility and toxicity limitations of BNIP bioactive agents. Last but not least, I would like to acknowledge my group, and in particular Zeynep, who is also within the audience, who has done all of this work with the parasites, although she was not very happy at the beginning to work with the parasites. Now she loves parasites and anti-parasitic research. And I would like to thank also my collaborators, both in Istanbul Medipol University as well as in Yeditepe University, and also the group of Ana Tomasz in Porto, who has provided us some experience with the techniques where Zeynep has visited the lab. And I would like to also thank our funding agencies, the Scientific and Technological Research Council of Turkey, and its project, which was integrated to a EU Cost Action Project, CM1307, entitled Targeted Chemotherapy Towards Diseases Caused by Endoparasites. And as we are in the Switzerland, each time when I come here, I feel myself more optimistic and more positive with the research on nanomedicine. And I would like to thank you all for listening. And if you visit my beautiful city, please let me know so I can be your guide. Thank you very much.